Hello, this is Ed Chapman, and in this video cast, we're going to wrap up the function and structure of the plasma membrane, otherwise known as the cell membrane, and we're going to focus on bulk transport, otherwise known as endocytosis and exocytosis. And the best place to start is to remember, or at least to attach to bulk transport, the need for energy. Just like with active transport, bulk transport does require the cell to spend some of its ATP energy reserves to make happen. Also, bulk transport involves a gross change in shape of the plasma membrane. So this means it also is going to be involving the cytoskeletal elements that allow all cells to move. And this is going to be controlled or regulated by the types of receptor proteins that are embedded in the cell membrane, which act as target recognition systems, so that the cell has a way to, quote, know, unquote, what it's endocytosing or what it's surrounding. Bulk transport is the way that cells move large amounts of material into or out of themselves. And the two most familiar words associated with this are endocytosis and exocytosis. And the type of endocytosis that you're going to probably be asked the most about is phagocytosis, which is a cell surrounding material that it considers food or that it has detected and determined is food, and placing this material inside of a food vacuole that's formed by the cell membrane that surrounded it. Exocytosis is a similar process, and we saw this in the Golgi apparatus excreting vesicles which make their way to the cell membrane and get excreted to the outside. So it's a matter of direction. Endo is in and exo is out. And just remember, phagocytosis is a kind of endocytosis. And cells that do this are called phagocytes. And in humans, that's a type of white blood cell. All right, so let's look at a picture. There's a lot going on here that you really don't need to know much about, but the basic idea is this. In phagocytosis, there are membrane-bound receptor proteins, represented here by these little shapes, that are able to detect a food particle, okay, which is this little green guy here, and stimulate the cell membrane to change its shape by altering the structure of these microfilaments, which are a protein called actin. And the end result of that is you get this food particle surrounded by cell membrane material. And this is now a vacuole. Specifically, it's a food vacuole. And this can then bind with lysosomes and all the other things we talked about earlier to get digestion accomplished intracellularly. There are other ways of doing this over here. To be honest, I know very little about this, but the idea is the same. Phagocytosis is the movement of material into the cell by the cell changing the shape of the membrane. All right, thanks for listening, and that wraps up our study of cell membrane structure and function.